Hello, and welcome to my endomembrane project. So before we start, I just wanted to clarify a few things. Number one is that everything here was drawn on Krita, so here's my watermark for what it's worth, so you know I drew it. And it was also supposed to be animated on Krita, but the exporter didn't work, so I just had to resort to screenshotting each individual frame and then pasting them together here in Adobe. And I'm telling you this because that's also why I had to split this video into four parts because there's just so many frames that my computer wouldn't like it if I put them all into one big video. So just to make things easier on my computer. So this video is going to be for scenario A, and scenario B, C, and D will all have their own videos, and I'll put them in a playlist on YouTube. So let's talk about the endomembrane system. So endo meaning inside, in this case inside the cell, and membrane meaning, well, the membranes that make up the organelles that make up the system. So the membranes are phospholipid bilayers, and I drew a little diagram there. It's just two phospholipids that make up the membrane. And this system carries out a few tasks, mostly the synthesis, modification, and transport of proteins, as well as the movement of lipids, the detoxification of poisons, and metabolism. But for this project, we're going to focus mainly on the first three tasks, the transport, modification, synthesis of proteins. This system includes the nucleus, both endoplasmic reticulums, the Golgi apparatus, the cell membrane, ribosomes, lysosomes, and various vesicles and vacuoles. All four scenarios of this project involve proteins that are synthesized in the rough ER, so they all start the same way, which is as chromatins in the nucleus. And chromatins are basically strands of genetic information that consists of DNA wrapped around histone proteins. And you guys aren't here to learn about protein synthesis, so I won't get too deep into that. But the gist of it is that DNA is a double helix and it gets unzipped by enzymes, so you end up with two sister strands. And one of these strands is used as a template to transpose RNA, which is then sent out of the nucleus and into the rough ER. The RNA cannot exit the nucleus by itself because the nucleus is a double membrane. It has two phospholipid bilayers, so four layers of phospholipids in total, and it needs the help of a pore complex, which is an intricate protein structure that perforates the nuclear envelope to help it get through. And these pore complexes are important not just in the entry and exit of proteins and RNA, but also large macromolecules as well. Yet now the RNA has exited the nucleus and is ready to enter into the rough ER. And this is how all the scenarios are going to start off. And speaking of which, let's talk about scenario A. So for scenario A, we're supposed to demonstrate the formation and pathway of a secretory protein that needs to be released from the cell. So the rough ER isn't very detailed in this picture, so I drew a closer look at it to really show the cisternae, which are the membranous sacs that have ribosomes attached to them, and these make up the rough ER. So once the RNA exits the nucleus, it is translated by one of these bound ribosomes into a polypeptide, which is then threaded into the inside of one of these membranous sacs called the ER lumen, and then it is going to move through these cisternae, and as it moves it's going to fold into its functional protein shape. And what you're going to see here is going to look like it's just teleporting from one cisterna to another, but that's not what's happening because in a real cell they're all interconnected, but that would require me to draw a 3D image and I didn't know how to do that, so just know that they're not teleporting. Okay, they're interconnected, so that's how they can move like that. Okay, so as they move from one sac to another, they're folded into their shape until they eventually reach this specialized region called the transitional ER, which is another membranous sac, but this one lacks mostly in ribosomes. And it is here that the protein is budded off into a transport vesicle, and the transport vesicle is just the membrane that contains this protein, and it is in this vesicle that it's going to get shipped out from the ER into the Golgi apparatus to be modified. So after this vesicle has left the rough ER, it's going to head towards the Golgi apparatus, which is also made up of a bunch of membranous sacs, the cisterna. So the membrane of the vesicle will fuse with the membrane of the Golgi, and this will inject the protein into the Golgi apparatus, and the membrane of the vesicle fuses with the membrane of the Golgi. So the Golgi is unlike the rough R in the sense that its cisterna are not interconnected, they're individual. So they move from a cos to a trans direction. And as you see this protein move from sac to sac, it's actually in the same membranous sac the whole time. The sac itself is just moving from the cos to the trans direction. And once it has reached the end of the trans direction, it is once again butted off into a transport vesicle, which is then sent to the cellular membrane. Oh, and the protein is modified as it moves through the Golgi apparatus. So now the finished product, the modified protein, is sent in the transport vesicle to the cell membrane, where the membrane of the vesicle once again fuses with the cellular membrane, and that ejects the protein out of the cell. And the protein of the vesicle fuses and adds to the cellular membrane. So there you go. There's part A.